Welcome back to Sync Study. We are going to study cytokinesis or cytoplasm division in this video. This is the continuation of mitosis cell division of previous video. In previous video, we have studied 4 phases of karyokinesis or nucleus division of mitotic cell division. So cytokinesis occurs after telophase stage. In telophase stage, 2 nuclei are formed at 2 poles. After the formation of 2 nuclei, there should be division of cytoplasm in order to form 2 daughter cells. Okay, so cytoplasm division occurs in cytokinesis, right? So in animal cells, cytokinesis takes place by cleavage method, whereas in plant cells, cytokinesis takes place by cell plate method. Okay, so we'll see how it happens in animal cell. Animal cell is surrounded by thin flexible plasma membrane. So plasma membrane is thin and flexible. That is why it can be pressed inward. Okay, that is what happens in the middle of the cell from 2 sides, right? So there forms a constriction, a little bit deepened area in both sides, and this constriction or cleavage is both of this from both the side. It starts moving inward. This keeps moving inward until they meet with each other somewhat at the center of cytoplasm. Okay, so plasma membrane breaks where these meet with each other at the point where 2 cleavages meet with each other, and the cytoplasm gets divided into 2 parts. Each part will be containing a nucleus, right? So this is cleavage method. So here forms 2 different cells, 2 daughter cells from a single cell. Okay, in animal cell. Now what we have seen in cleavage method is, in animal cell, cleavage is forming from outward to inward. In the beginning it appeared on the outside wall, then it started moving inward from outward to inward. Okay, that is how cleavage is formed in animal cell, outward to inward. Whereas in plant cell, cytoplasm division takes place by cell plate method, and plant cell is surrounded by rigid, hard cell wall. Okay, so cell wall cannot, because of this, cell wall cannot be pressed inward. That is why a small cell plate starts forming in the middle of cytoplasm, right? So Golgi complex helps in this method actually in forming cell plate. So cell plate appears in the middle of the cytoplasm, and this cell plate starts growing outward from 2 sides, from both the sides. It keeps growing in both the sides until it meets cell wall. Okay, when it meets cell wall, it breaks at that point, dividing cytoplasm into halves, right? So each half is containing a nucleus in them. So this is how 2 daughter cells are formed from a single mother cell. Okay, so in case of plant cell, in cell plate method, what we have seen is cell plate is formed from inward to outward. Okay, it appeared in the middle in the beginning, then it started growing outward. That is why cell plate formation takes place from inward to outward, inward to outward. Okay, so this is the method by which cytokinesis takes place in animal cell and plant cell. So these are the differences as well. In animal cell it is cleavage method, in plant cell it is cell plate method. In animal cell, cleavage forms from outward to inward. In plant cell, cell plate forms from inward to outward. These are the differences. And at the end, what we have seen is 2 daughter cells are formed from single mother cell in animal cell, and in plant cell also 2 daughter cells are formed from a single mother cell. So this completes mitosis cell division as well as cytokinesis division. We are going to study meiosis in next video.